أهلا بكم في دروس الأحياء للصف الثالث ثانوي لغات. Today's lecture will continue the questions and answers of the book. So let's begin. Question 6, state the site and the function of the thymus gland. The thymus gland secretes thymosin hormone that stimulates maturity of lymphoid stem cells to T cells, and it's situated in the chest, anterior to the trachea. Pardon me. Site and function of the spleen. It's in the abdominal cavity, which is the البطن يعني, in the upper left corner, and next to the stomach, and it has a lot of specialized white blood cells called macrophages that pick up all that is strange about the body and disposes of the senescent or the old blood cells as well. Okay, you know that the blood cells have a lifespan of 120 days, and the spleen's function is to get rid of the old RBCs. Contains also lymphocytes. Site and function of the tonsils. They pick up any microbe or foreign body entering the digestive or respiratory tract because it's in the mouth. Peyer's patches, the real function is unknown, but they have a role in immunity in the body. Natural killer cells, they attack the body cells infected with viruses, carcinogenic cells, or transplanted organs. Cerumen, it's the earwax, and it kills microbes and protects the ears. Okay, the diagram, the opposite figure, label the opposite figure. This is the variable site of the antibody. 2 is the constant site of the antibody. 8 is the heavy chain. 6 is the hinge ده المكان ده اللي هو complement binding site اللي هو 7 complement binding site, and D disulfide bonds اللي هي connects the heavy chains مع light chains, وده اللي هو الانتيجين antibody او antigen binding site. اوكي, دي بالنسبة للليبلز على الفيجر. What are the heavy chains and the light chains and how they bid to each other? Each antibody consists of 2 pairs of polypeptide chains. 2 of these chains are long and called heavy chains, while the other 2 chains are short and called light chains. They bind to each other by disulfide bonds. How antibodies differ from one to another. According to the conformation of amino acids that differ in their sequence, types, and shape at the antigen binding site. Okay, what is meant by the constant and the variable regions of the antibody? The variable regions is it's an antigen binding site where each antibody has 2 identical antigen binding sites. Shape of the site is different from one antibody to another and varies from antibody to another. The constant region, however, is the next part of the antibody, the rest of the antibody, a constant shape among all types of antibodies. Okay, how antigen antibody complex is formed. Antigen binding site helps for the binding between the antigen and specific antibody. Okay, the inflammatory response is produced when a body cell is harmed. What is the role of histamine in the inflammatory response? Histamine increase the permeability of arterioles and capillaries to blood fluids, and then leak from the blood circulation, leading to swelling of the tissues, allows the passage of chemicals that kill and dissolve bacteria at the site, and enables white blood cells as neutrophils, monocytes, and macrophages to fight and kill foreign bodies and microbes. What is the benefit obtained from the involvement of different types of white blood cells? It increases the capacity of the or the immunity for killing different pathogens. Okay. Identify the role of memory cells in protecting body from disease. Memory cells during the first immune response, when the immune response to the new pathogen, the two types of memory cells are that means B and T lymphocytes are produced, which store information about the antigens formed by the immune systems in the past. The first immune response, or the primary immune response, and they can live for tens of years and may survive till death. During the secondary immune response, when the same individual is infected with the pathogen again, the memory cells respond to the pathogen once it enters the body and kills it once it enters the body, without leaving it a chance to demonstrate the symptoms of the disease. Okay, mention some of the natural immunity mechanisms that represent the first line of body defense in man. Remember the barriers: the skin with its horny layer and sweat glands. Sweat in the skin also helps. Cerumen as the earwax. Tears for protecting the eye. Mucus in the respiratory tract. It's viscous fluid, so adheres to any foreign body. Okay, to be expelled to the outside. The saliva, for example, it has enzymes and that dissolve such microbes. Acidic gastric juice, with remember, it reaches 2 pH. That can kill many microorganisms in the food we eat. Okay. Mention the morphological changes that take place in the plant cells after being infected by microbes. This is known as the cellular immune structures, such as swelling the cells or the cell walls of epidermal cells and the cells under the epidermis during the direct penetration of the pathogen. Its function is to inhibition of the penetration process through the infected cells. Also surrounding the mycelium, which attacks the plant with an insulator cover. Its function to prevent the transmission of fungus from cell to another. Okay, mention 3 lymphatic organs that play an important role in the immune system in human body, in the human, and then explain the role of each organ in protecting the body. Tonsils, for example, they pick up any microbe or foreign body that may enter the food with food or air and prevent its entry to the body. The thymus gland, for example, secretes thymosin hormone that stimulates maturity of T cells. The bone marrow, another example, is responsible for the production of red blood cells white blood cells, and blood platelets. Okay, mention this we answered that. Explain by the help of labor diagram transverse section in lymphatic node. You should have learned this, how to draw it. Remember the afferent vessels on the convex border, and the efferent vessel is from the concave border. The structure of the antibody, remember the two light chains and heavy chains, disulfide bonds, the complement binding sites, the variable region, the constant regions. All this you must label on drawing of the antibody. Okay, 15. Explain the different mechanisms of antibody action. Remember, we discussed this before in lectures. One of the mechanisms is neutralization. It binds to the virus, and this binding will prevent the virus from adhering to the membranes of the host cell. For example, agglutination, where the antibody binds to more than one microbe, like the immunoglobulin M, for example. Lysis, where the antibody binds to antigen then that activates complements to lyse the coats of the antigens. Precipitation, where the antibody binds to antigens to form insoluble precipitate, and I told you this happens with the soluble antigens to turn them into insoluble precipitate. Antitoxins, for example, where antibody binds to toxins to activate the complement that leads to detoxifying toxins and makes them easily to engulfed by phagocytosis. Okay, describe how lymphocytes identify pathogens and they bind to them. During humoral or antibody-mediated immunity, the B lymphocyte recognizes and attaches itself to the antigen by using immune receptors present on its outer membrane or outer surface. The antigen binds to the major histocompatibility complex, then the complex resulting from the binding between the antigen and the MHC transfers to the plasma membrane of the B lymphocyte to be presented on the surface. During cell-mediated immunity, this involves T cells or T helper cells lymphocytes, which has receptor CD4 on its membrane, will recognize the antigen by its binding to the antigen MHC complex that presented on the surface of the macrophages. This concludes answering the book questions. I hope you got the maximum benefit from this lecture, and thank you very much for watching.